Thanks so much, Renee. It's really a pleasure to be here today. I'm going to share some fully unpublished work with you all today. So this is hot off the presses and not available anywhere but this Mount Sinai feed. As Renee mentioned, my lab is very interested, at least part of it is interested in identifying the role that certain brain circuits play in stress susceptibility and stress resilience. And I'm sure I don't have to remind people that last year has been a particularly stressful and trying times. I think probably about a year ago from today, we were all in this mode, thinking about where we might find our next roll of toilet paper. And that quickly then led to the realization that we were home with children and trying to work and maintain our labs, maintain our jobs, maintain some semblance of normalcy. It also was associated with significant social isolation. And then we learned, I think, early on in the pandemic that the virus itself, if you were unlucky enough to have contracted it, could have some long-lasting neuropsychiatric effects and partly due, or at least it's thought, due to changes in the way in which the virus is acting on the immune system and how that affects your brain. But there's another type of stress that ramped up really significantly and in a very concerning way, and this is interpersonal violence. This is a perspective written in the New England Journal where they called it a pandemic within the pandemic. This was particularly difficult for women, but across the spectrum, we saw increases in interpersonal violence, probably because we were stuck in the confines of four walls together for very long periods of time and were stressed out. But just to show you some data, this is a study from the US showing rates of nearly, upwards of 10 to almost 30% increases in interpersonal violence that we saw last year. This wasn't isolated to the United States. Data out of Europe and South America show similar rates of increases, upwards of 25 to 30%. And what is this type of increase in interpersonal violence? What is this going to do to the mental health of our population? Well, it's not quite clear yet. I don't think we've completely rounded the corner on the pandemic, and so the numbers are starting to trickle in. We've certainly seen a significant rise in neuropsychiatric events. But if we go back to the literature and look at the impacts of such interpersonal violence, it's quite dramatic and concerning. So this is a study looking at the activity patterns in brain regions that control our responses to rewarding and pleasurable events. And they found that if you experienced relational victimization in the past year, that these brain regions weren't functioning as they were normally, and ultimately it was impairing your ability to experience rewarding events in your life. Similar studies in children have shown that victimization or violence towards them, or even just childhood maltreatment in general, can lead to an increase in the child's reactivity to threatening stimuli, suggesting that they experience aversive and threatening stimuli in the environment with a far more robust response. So that led us to think about what this might mean. Our hypothesis going into these studies was that negative social experience, one that might be experienced through partner violence or aggression outside of your immediate personal relationships, may actually lead to a heightened activation of brain regions that are typically involved in our perception of stress or threat, and that activation of the overactivation or inappropriate activation of those stress circuits in times when they shouldn't be on may actually occlude our ability to experience rewarding events. And one in particular is social behavior in and of itself. So our ability to experience social behavior as rewarding, and our desire to engage in social interaction. And that ultimately this may lead to severe social avoidance behavior. So to get at this, we use mouse models because it allows us to better interrogate neural circuit mechanisms to define functional circuits that may control such a response, but also to test novel targets, either at the drug level or at the neuromodulatory level, to determine circuits that we might tap into to promote resilience to such a social stress, thereby kind of reversing these maladaptive behavioral responses. So to mimic violence or aggression, we use a social defeat stress model. This is an established model of stress. We select large, aggressive mice that are screened for their for being particularly aggressive and their willingness to subordinate a broad range of social targets. We can do this in both males and females, but in both of our models, the aggressor is always a male mouse. So we then introduce a smaller, more subordinate C57 black 6 mouse into the cage with this aggressive bully. They are socially subordinated for 5 to 10 minutes each day over a 10-day period. This is a very chronic and consistent social stress that they experience. During the remainder of each day, they're housed on the other side of a plexiglass divider. So it's thought that they're experiencing both the physical stressor, but also a significant psychological stressor. We can then test their social behavior and determine whether they're susceptible, meaning they show a particular behavioral phenotype, or whether they're resilient and have managed to resist the development of any significant behavioral maladaptations. We do this first by screening social interaction, placing a novel CD1 mouse into a goal box here, and then allowing the experimental C57 mouse to interact with that mouse in a goal box. Mice that interact normally or similar to controls, and the quantification is down here, we term them resilient. Mice that avoid the social target when they're present in the cage, we call them susceptible. It's important to note that susceptibility and resilience are not black and white terms. You can be susceptible to certain features of the stress and resilient to others, and vice versa. But in general, we define susceptibility as a loss of social interaction that's associated with a host of physical abnormalities, including a type 2 diabetes-like effect and changes in the peripheral immune system indicative of chronic inflammation. We don't really know exactly what drives susceptibility and resilience. These experimental mice, it's important to note, are genetically identical. They're inbred strains, and so the difference between coping or susceptibility and resilience is not defined by some genetic code like it might be in humans, but rather we think it's due to either behavioral or other related phenomenon that either allow the animal to adapt in one way versus the other. And so one of the things that we've begun to look at is behavioral coping strategies. What is the experimental animal doing in the cage while they're interacting with this large aggressive CD1 mouse? We've identified 3 behavioral coping strategies, which we've termed passive, active, or outright fighting back. And these types of behaviors can be quantified during the social defeat episode. Passive coping is when the animal kind of gives up. It's maybe more akin to learned helplessness behavior. They throw their arms or their paws in the air and say, "I'm done, you win." Active coping is when the animal actively tries to avoid such social subordination, jumps and hops away from the aggressor and runs around the cages. And then fighting back is exactly what it sounds like. They put their dukes up and push the aggressor back and try to limit the amount of aggression that they experience. And in fact, if you quantify these coping scores over time, you can see that actually the degree to which the animal is socially avoidant correlates with coping, with the higher coping scores reflecting the more active approaches of running away and fighting back, and the lower coping scores reflective of these more passive responses. So it does seem, at least at some level, that it's a behavioral strategy. Now, what are the kind of brain circuit mechanisms or underlying mechanisms that drive this is something we're quite interested in. But I do want to highlight one thing. we've used this model over the years, and we've interpreted these reductions in social behavior as being a loss of social reward or an inability to experience social interaction as being pleasurable. But in reality, with these types of paradigms, I think it's really difficult to make those conclusions. And the reason is that, as I mentioned here, our output, our test is looking at interaction with this CD1 mouse. Now, regardless of the fact that it's not an aggressor that it's seen before, that it's novel, that it's never actually been attacked or subordinated by it, mice generalize cues. And it's very likely that the avoidance of this target is based on fear of the traumatic experience that it had with one mouse that looked very similar to this. So I don't think these types of behavioral assays really tap into this idea or this construct of social reward and anhedonia. So to do that, we've established a slightly different set of derivations to the model. We still do the chronic social defeat stress the same way we've always done it. We still define susceptibility and resilience as the interaction time spent with this novel CD1 mouse, but we now test social behavior in the presence of a non-threatening, same strain, same sex juvenile mouse. This is a 3 to 4 week old pup. So non-threatening in any way, shape, or form. Under control conditions, 3-week-old pups are typically rewarding to adult male and female mice. And so we think that by looking into the types of interactions that occur under these circumstances, we may better tap into social reward processes. So in fact, when we do this new test of juvenile social interaction, we still see that susceptible mice show reduced investigation time with the juvenile, and they show more bouts of active avoidance. Active avoidance is defined when the juvenile approaches the adult mouse and the adult mouse retracts and runs away. So this does seem to impair social behavior even to non-threatening social stimuli. But to get at reward preference, we use a slightly different model. This was borrowed from the drug addiction literature. It's a conditioned place preference apparatus that's been used historically to measure the rewarding properties of all drugs of abuse. In the model, we have contextually distinct chambers defined either by these white and black stripes or gray zones. The contexts differ by tactile cues as well as visual cues. This is really so that the animal can determine and learn environments associated with a certain cue. In the case of drug addiction, what we would often do is inject a rewarding dose of a drug of abuse into one of the two chambers, condition the animal 30 minutes a day for 4 days, and then by the end of that 4 days, you put the animal back into the box, allowing them free access to both conditioned chambers, and you test to see where their preference is. And for all addictive drugs of abuse, their preference is toward the side in which they receive the injection of the rewarding dose of the drug of abuse. We replace the drug of abuse in our model with a juvenile, same sex, same strain mouse, and we do the conditioning in a similar fashion. And then at the end of that conditioning session, we again place the animals back into the box and we look to see which side of the chambers they like to spend their time in. Both male and female mice spend more time after training in the side in which they were allowed to interact with that juvenile male mouse, and that's shown in these red dots up here on both sides. So male and female adult mice, this is under control conditions, both find these juvenile C57 mice to be rewarding. Following social defeat stress, we see a different pattern. So again, we repeated the social preference that we observe in control mice. These are mice that did not experience any significant social defeat stress. Same phenotype is observed in resilient mice, but susceptible mice fail to develop this conditioned reward response, and they do not distinguish between the social paired chamber and the social unpaired chamber. They don't find it aversive either, and I think that's an important point. For example, if you were to put an animal into one of these chambers with a rewarding dose of opiates, and then hit them with an opiate antagonist, they don't just prevent reward to that opiate, they actually produce aversion because it's an aversive internal state. So we're not seeing an aversive internal state, but what we're seeing is a blockade of the ability of these animals to develop conditioned reward to these social targets. So to get at the brain mechanisms, we took a more high altitude approach to start with. We wanted to get a general sense of what brain areas were becoming activated in response to a juvenile social target in control susceptible and resilient mice following the 10-day social defeat stress paradigm. So we used a brain clearing procedure that allows us to clear the brain tissue out so that it's fairly transparent, and you can then stain for markers of cellular activation throughout the entire extent of the brain tissue. In this case, we use CFOS, which is an immediate early gene that's activated in response to neural activity. So it's a good broad marker to look at neural activity in response to a particular stimuli. We sacrificed mice following, as I said, 10 days of social defeat stress and a juvenile social interaction. So we're really looking at CFOS that's being induced in response to that juvenile social interaction. And what you can see as you look three-dimensionally through the tissue sections, you can see nice staining of CFOS. As we zoom in, you'll be able to see it better, but these are small circular dots that you'll see throughout. And each one of those is associated with a specific cell type in the brain. And then it allows us to kind of quantify the number of those cells that we see in various brain regions and throughout. Okay, so this is a table just showing the brain regions that came up. You can see entorhinal cortex came up as strongly activated in susceptible mice compared to control and resilient, the lateral septum, and a whole host of other structures. This table is from male mice. The male and female brain circuits that were activated in response to this are quite different. But we did note that the lateral septum, which is one of our top regulated regions, was among the very few that were similarly regulated in males and females. Given that we saw similar behavioral phenotypes, we wanted to explore the lateral septum and its target regions in controlling some of these maladaptive responses to social stress. So we found within the lateral septum that a specific type of cell that expresses the neuropeptide neurotensin and then also the inhibitory neurotransmitter GABA was the cell type that was predominantly activated in response to these social targets. So in the left-hand panel, I'm showing you in situ hybridization images in control, resilient, and susceptible mice. Again, this is following 10 days of social defeat and a juvenile social interaction test. We normalize these data to animals that go through 10 days of social defeat without the juvenile interaction test. So we're measuring CFOS that's being activated by that juvenile social interaction. CFOS is in red, neurotensin is in green, and you can see that susceptible mice have strong activation based on CFOS expression within these neurotensin cells in the lateral septum. And we see similar responses in male and female rodents. Resilient mice, though going through the stress, do not seem to activate these cell types in response to the interaction. So we wanted to dig a little bit deeper into this cell type. There wasn't a lot known with relation to stress. The lateral septum itself is really thought to play an integral role in interfacing between, for example, memory circuits and reward circuits, and has been shown to play roles in the response to reward, but we didn't know anything about these LSNT neurons. So interestingly, so the way that we did this was to use an in vivo approach so that we could look at neural activity in real time as these mice were interacting either with a social target, a stressful event, or some other stimuli with a specific valence tag to it. So we used the calcium indicator GCAMP6. We did this by injecting an adeno-associated virus that was Cre-dependent, meaning that it only expresses in cells that also express the enzyme Cre recombinase. We did this into an animal that was engineered to only express free recombinase in those neurotensin cells, which means that we can now measure activity, calcium activity, specifically within the neurotensin cells in the lateral septum. Calcium activity is a surrogate marker of neuronal action potentials, as it's required for all nerve cells in the brain to fire. So that by measuring changes in calcium activity during this social interaction, we can now hypothesize or conclude the activity patterns that are associated with certain behavioral events. So we put the animals through chronic social defeat stress. We test for susceptibility and resilience. We then measure activity of LSNT neurons during juvenile social interaction. And you can see that these cells don't respond in control mice to the juvenile. Now, this is a rewarding event for these animals, and so it does kind of argue that these cells are not necessarily playing a role in sensing reward or responding to rewards in the environment, at least in unstressed control mice. In resilient and susceptible mice, we see a pattern that's quite different from one another. Resilient mice, again, look very much like controls. These cells do neither activate nor inhibit in response to a juvenile social target. But susceptible mice have a very strong activation each time they interact. These red ticks at the top reflect social bouts with the juvenile. And you can see that the calcium activity goes way up in response to juvenile interactions. And this is just the quantification of the total amount of fluorescence emitted by the calcium indicator over the entire session of social interactions. So it does appear that these cells might be sensing something more akin to threat or stress, but we're not quite sure at this point. So in order to get at that question a little bit more, or to examine that question a little bit more closely, we wanted to remove the social context and add in non-social threats or stressors and see how these neurons respond. So we did two different ones. One actually was a social threat, the other was a non-social threat. The social threat was a CD1 aggressor attacking it, and you can see that these LSNT neurons respond very robustly to attack signals, almost though similar to a juvenile, which is quite shocking that these cells now activate to levels comparable to when they're being attacked by a CD1 mouse. So that tells you how stressful the juvenile social interaction must be, or just maybe general interaction. We also did an additional stress where we hung the animals by their tails, and you can again see very strong activation of these circuits. Interestingly, rewarding stimuli do not activate or inhibit. So again, these cells seem to be coming on in response to threat or stress. To determine the functional relevance of these cells in terms of modifying this behavior, we used a chemogenetic approach. This is a synthetic receptor that responds only to a synthetic drug. And so this allows us to either activate or inhibit these cells. I'll just show you how we did this. We chose previously susceptible mice, and we then inhibited these LSNT neurons. And by doing so, we can bring social interaction way up, comparable to those that we see in resilient mice. And we can then promote a more normal social CPP. We can take previously resilient mice and do the opposite, where we can now promote social avoidance by activating the LSNT neurons and then occluding social reward. I think I'm going to skip this last part in the interest of time. I'll just say with this that we're now starting to probe circuits downstream or upstream of the lateral septum neurotensin cells to see how they may be occluding social reward. Our hypothesis is that these cells are projecting to primary reward centers like the nucleus accumbens and nucleus of diagonal band, and that by direct communication, they're promoting or opposing social reward behavior. So they're occluding social reward in the context of susceptible mice. And with that, I will just thank people in my lab that did the work. In particular, I'd like to thank Long Lee. This work was led by Long, Catherine, Roman, Joey, and Tony. and the rest of my lab and our funding sources, of course. And thank you all for your attention. I'm happy to answer questions.
Thank you very much, Scott. That was a great, very original, great studies. You're right. We have time for 1 or 2 questions, and people can use the Q&A box, which should at the bottom of your screen. And then you can type in the question, and Scott Russo will then read and answer the question. So please go ahead with the Q&A. In the meantime, Scott, I do have a question. What do you think is the main characteristic between the resilient and the susceptible mice? I mean, if you would put your money, they're all genetically identical, I think. So that's a little bit difficult. But I mean, where do you think is the main part of the answer?
I certainly think there's developmental issues that we don't control for, that we can't really define coming into it. So there's certainly, neurobiological differences based on how they develop. At a behavioral level, we kind of think about it as them taking a particular strategy. Individuals are faced with decisions all the time, and you have to choose what your strategy is going to be in life. And I think it can be learned. In fact, the coping that I showed you early on is a learned event. We don't see those differences at the early stages of defeat. So I think maybe it's serendipitous. Maybe an organism decides how to respond in real time during a threatening event. And if they respond one way, they learn to then continue to respond in that way. But again, in humans, it's quite more complicated, right? I think there's going to be genetic factors, developmental factors, and others. But
yeah. So Scott, in the meantime, we have 2 questions in the Q&A. If you, actually 3, if you can address them,
Yep.
Because we'd like to have them all 3 answered pretty briefly.
Sure.
That would be great.
So Lana wants to know about the coping strategies. They reminded her of behaviors that they observe in humans associated with attachment styles. Wondering how you think of early life experience. I mean, it's a fantastic question. What we can do in a controlled fashion is change early life experience. So we can change the maternal relationship, periods of maternal separation, for example, enhance these processes so they promote resilience. So I think much like humans, early experience is going to be critical. And we're starting to kind of think about that in the context of reward as well, although we don't have a final answer. The second question is similar, and it's related more to the epigenetic landscape. Again, we haven't looked at the epigenetic landscape of the lateral septum and T neurons. My guess is that we will find broad differences between susceptible and resilient based upon published work from Eric Nestler's lab across other brain circuits. And again, I think this will be defined a lot by those early life experiences, that the epigenetic switch is determined by those early experiences, and they then change how you respond as an adult. And the last question is related to neuromodulators in stress resilience, for example, dopamine and serotonin. It's very complicated. I'll speak just to dopamine because I think in the interest of time, we probably should move to the next speaker. And in fact, it's a great segue to the next speaker, who's going to be talking about testing novel therapeutics that are based on altering the physiology of dopamine neurons. And the work was conceived at the preclinical level using the social defeat stress model. Dopamine is complicated. So actually, increased firing of dopamine neurons is associated with increased stress susceptibility. And if you can silence those dopamine cells from firing to the extent that they do, it actually promotes resilience. So it's food for thought. I don't think it's through a dopamine specific mechanism. I think there's other mechanisms involved, other neuromodulators in dopamine cells. But James is going to really, I think, dig into that in the next talk in the context of human depression. So stay tuned.
Yeah. So Scott, I mean, there's one last question that you can just answer with yes or no. So that's a very easy one. Can you see it?
Oh yeah. Sure. Like drugs, are mice capable of experiencing extinction to social fear? Yes, and actually the susceptible mice don't extinguish as quickly or as readily. So.
All right, great. Scott, thanks again. Thanks so very much. And great, we could address all the questions.